Welcome to the SANS Science Experience podcast, where we explore the latest and most exciting papers in preprints in science. Before we jump into today's molecular deep dive, please take a moment to hit that like button, subscribe if you haven't already, and maybe share this deep dive with your friends and family. It really does support the SANS Science Experience. Okay, so today we're embarking on a mission sort of to understand what might be the ultimate biological time capsule, circular RNAs, or circRNAs for short. Now, if you think about the traditional linear messenger RNA, mRNA, that's like the quick disposable instruction manual from DNA, right?
Right. It does its job and gets degraded pretty quickly.
Exactly. But these circRNAs, they seem to be something else entirely.
They really are. They're these incredibly stable RNA molecules, and they're made through this unusual process called backsplicing.
Backsplicing. Okay, for listeners maybe less familiar with the cell's machinery, what exactly is that? Why does it make circRNAs so special?
Well, it's basically about the shape. Normal RNAs align, right, start to finish. Easy for enzymes to find an end and start chewing it up.
Makes sense.
But with backsplicing, the cell's splicing machinery, which usually cuts and pastes RNA segments together, linearly kind of, loops back on itself. It forces the end of an RNA segment to covalently link to its beginning.
So it bites its own tail, essentially, forming a closed circle.
Precisely. A closed loop, no ends for those degradation enzymes to easily grab onto. And that covalent closure is what gives them this exceptional stability.
Okay, super stable. And the paper mentions they're highly enriched in the brain.
Very much so. Which is interesting in itself. And this paper we're looking at today, Curio et al., 2025 in Cell Reports, really zeroed in on the stability aspect. They made this, well, pretty stunning claim that circRNAs don't just exist, they pile up dramatically as we age.
Accumulate with age. Okay.
And more than that, they might act as these long-term biological flight recorders, like a logbook for experiences an animal has had.
Wow, okay. A flight recorder. That's the big idea we're diving into then. So the mission of this study, what were they trying to figure out specifically?
Their main goal was to understand why. Why do these circRNAs increase so much with age in the brain? Is the cell making more of them as it gets older?
Or maybe it just gets worse at cleaning them up.
Exactly. Production versus degradation. To figure this out, they used Drosophila, the common fruit fly, and looked at the circRNAs in their heads across their entire lifespan.
Fruit flies. Classic model. Okay, let's get into what they found. Were circRNAs actually good age markers?
Right, that was step one. Establish if they're reliable timekeepers. We know lots of linear mRNAs change with age, but it's often messy, inconsistent.
Yeah, finding a clean biological clock is tough. So what did the circRNAs show?
They showed incredibly linear accumulation. They measured the circRNA levels at 6 different time points, from flies that just emerged from their pupa.
Newly closed, yeah.
Right, newly closed, all the way up to 50 days old, which is quite old for a fly. And the correlation with age, it was just astonishingly tight.
Okay, you're saying astonishingly tight. Biology results are often noisy. How good are we talking, like R-squared values?
We're talking precision you almost never see in biology. The R-squared value for the linear regression, how well the data fits a straight line against age, was 0.99 for female flies.
0.99, that's basically a perfect line.
Almost perfect. And 0.97 for males. It means the amount of circRNA in that fly's head is an incredibly accurate predictor of its chronological age, day by day.
Wow. Okay, that level of linearity is remarkable. It suggests the process is really robust, right? Almost mechanical.
It seems so. Very resistant to biological noise.
So what about the comparison? The standard linear mRNAs we usually look at. How did they fare as age markers?
Oh, a stark contrast. When they looked at the total linear mRNA reads, the changes with age were, well, all over the place. Inconsistent, often mixed up with other factors.
Like what?
Well, they used principal component analysis, PCA, that statistical tool that finds the biggest source of variation in your data.
Right, it tries to separate out the main factors driving differences.
Exactly. For the linear mRNAs, the PCA often showed that the biggest difference between samples wasn't age, but things like sex differences between the flies or just individual variation.
So confounded, not a clear age signal.
Not nearly as clear. They could maybe tell very young from very old, but not much in between reliably. But for circRNAs, the PCA robustly, clearly separated the samples based purely on age.
Okay.
It means you could reliably distinguish, say, a 10-day-old fly from a 30-day-old fly just by looking at their circRNA profile. Linear mRNAs often couldn't do that.
So bottom line.
Yeah.
circRNAs are far superior age markers. The data seems really strong on that.
Yeah.
Okay, so we have this near-perfect biological clock ticking away, accumulating linearly. The next big question is why? Is the cell factory working harder, or is the garbage collection just off?
It really seems to be the garbage collector taking a permanent vacation, not the factory speeding up. The study strongly concluded that this accumulation is driven almost entirely by their incredible stability, meaning very low degradation rates, not by increased production.
Right. And they had evidence for this. How did they disentangle those two possibilities?
They had three really neat lines of evidence. First, they did this clever comparison. Remember, circRNAs are spliced out of a longer linear RNA transcript, the host gene.
Yeah, the gene that produces both the circular form and the standard linear mRNA form.
Exactly. So the logic is, if the cell decided to ramp up the overall activity of that host gene, you'd expect to see both the circular and the linear versions increase together, right?
Sure, more raw material being made should mean more of both products.
But that's not what they found. They saw circRNA levels going up, up, up with age dramatically. But the levels of the corresponding linear mRNAs from the same host genes, they stayed relatively constant over time.
Ah, okay. That immediately suggests it's not just more gene activity overall. The circular form is piling up specifically.
Precisely. It decouples accumulation from just raw production increases.
Very clear. Okay, what was the second piece of evidence?
They looked directly at the factory activity. They measured the overall transcription levels of these host genes using a proxy, the signal from the intron.
Introns. The bits that are normally cut out and discarded during splicing.
Yeah. The amount of intron signal you detect is often a good measure of how actively the gene is being transcribed right now. And again, they found no overall increase in this general transcription signal with age.
So the factory isn't running faster. Got it. And the third piece?
The third piece ruled out changes in, processing efficiency. Maybe the cell just gets better at making the circles from the same amount of raw material as it ages.
Ah, like the backsplicing process itself becomes more efficient.
Right. But they checked for global changes in splicing patterns or things like intron retention across the genome and found the age-dependent accumulation of circRNAs didn't correlate with any of that. It wasn't a change in how they were made.
So it all points back to one thing.
Pure longevity, exceptional stability. They just stick around.
And this effect is particularly strong in neurons, you mentioned.
Yes, the age-dependent increase was predominantly seen in neurons, which makes a lot of sense because neurons are postmitotic, they don't divide, and they're very long-lived cells, especially in the brain. That's where these super stable circRNAs seem to be most enriched.
And when they actually measured the stability? The half-life.
This is where it gets, really quite wild. Their stability measurements showed circRNA half-lives exceeding 20 days. In vivo. In the fly brain?
Wait, over 20 days. The fly only lives for, 50 or 60 days total?
Exactly. A circRNA made when the fly is young could still be, substantially present almost halfway through its entire lifespan.
That's incredible persistence for an RNA molecule if you scale that to humans.
It's like finding a molecule in our brains that sticks around for decades. It puts circRNAs among the most stable molecules known in a living organism.
Just amazing stability. Which naturally leads to the next idea, the flight recorder. Right? If a molecule is that stable, it's not just a clock. It could potentially hold information for a very long time, like a diary entry.
That's the hypothesis they tested next. Can circRNAs act as markers of past life experiences?
How do they test that?
They used an environmental stressor, heat. They took adult flies, put them in a fairly uncomfortable temperature, 29 degrees Celsius, which is stressful for them, for 10 straight days.
Okay, a heat wave for flies.
Pretty much. Then, critically, they moved the flies back to their standard, comfortable 25-degree conditions and let them live out the rest of their lives, monitoring them for a long recovery period.
So a temporary stress early on, then back to normal. What happened to the circRNAs during the heat stress? Did they react?
Yes. They found certain specific circRNAs shot up significantly while the flies were at 29 degrees, like molecular stress signals being activated.
Makes sense. But the key question is, what happened after they went back to the comfortable temperature?
This is the kicker. Those specific circRNAs that were elevated during the heat stress, they remained significantly upregulated even 6 full weeks after the flies were returned to the normal 25 degrees.
6 weeks later. In fly time, that's a huge chunk of their remaining life.
It's a very long time. It's like detecting evidence of a bad fever you had years ago just by looking at molecules in your cells now. They carry a molecular memory, a scar of that transient stress.
Wow. So the memory persists long after the event is over.
It does. And when they modeled the decay rates for some of the most persistent markers, specific ones like circRL and circRF, the data actually suggested their half-lives might be effectively infinite in adult neural tissue.
Infinite. meaning they might just not degrade at all once they're made in those neurons.
That's what the modeling implied for some of them. They might be permanent fixtures once induced by an event.
So theoretically, you could take a fly that seems perfectly fine living in normal conditions, analyze its circRNAs.
And potentially tell that weeks or months ago, it went through a 10-day period of significant heat stress, even though the stress itself is long gone.
That absolutely confirms the flight recorder idea then. It detects transient environmental exposures long after the fact.
Yeah. Exactly. It transforms these molecules from just age markers into these enduring records of, well, life's hardships, essentially. A window into the animal's past environmental history.
And it's using this incredibly stable RNA mechanism, not just, say, epigenetic marks, which we often think about for long-term memory. That feels really novel.
It adds another layer, certainly. A very robust physical record. #tag conclusion and final thought.
Okay, let's try and synthesize this deep dive. We've learned that circRNAs, made by this backsplicing process, are extraordinarily stable.
Exceptionally stable, yeah.
They track chronological age with almost unbelievable linear precision, at least in flies.
Making them maybe the best molecular clock we've found so far.
And crucially, because of that stability, they can act as long-term biological memory recorders, flight recorders, capturing evidence of past environmental stress long after the event.
That's the core story. Stability drives accumulation, which tracks age and also locks in records of past events.
So what are the bigger implications here? What's the final thought for us to chew on?
Well, think about taking this out of the lab. If circRNAs track both age and these transient stress events with such permanence, you could potentially use them to read the history of wild animal populations.
Read their history. How so?
Imagine analyzing a population of wild insects, maybe fish, maybe even longer-lived animals. By looking at their specific circRNA profiles, you might get this integrated picture of their past.
You mean figure out their age distribution accurately?
Yes, accurately determine age structure, but also potentially measure their cumulative lifetime exposure to unpredictable stresses. Things like toxins in the environment, periods of drought, heat waves due to climate change, maybe even disease outbreaks.
So you could see the scars of those events on a population level long after they happened.
Potentially, yes. It could give us a historical record written into the molecules of how a population or even an individual has coped with its environment over time. A kind of integrated transcriptome revealing past struggles.
That truly is the ultimate biological flight recorder, a fascinating prospect. Well, thank you for joining us for this deep dive into the really remarkable stability and utility of circular RNAs. If you enjoyed hearing about these molecular flight recorders, please do remember to like, subscribe, and share this deep dive with your friends and family. It helps us out a lot. We look forward to sharing more exciting science with you soon on the SciENce Experience podcast.